E Claudio? Ready.
Okay. Bom dia, gente. Morning. So let's go for the second day of this house symposium. Welcome. So we'll have a first plenary talk. Today we will have the two plenary talks. One at this early morning, and the other one closing our house symposium. So it's with great pleasure that I'm going to ask Claudio to introduce Professor Pierre, and then we'll move on. Thank you, Claudio.
Thank you, Draulio. So good morning, everyone. Well, I'm just here to introduce Pierre Lupi. He's a long-time collaborator of the group. We're just doing some calculations. He's been around, our collaboration has been around maybe for 11 years already, at least, yes. So it's a great pleasure to welcome him and Monique Sclapes, that is also here. They are from France. Monique will speak in the afternoon. So to introduce Pierre, what can I say about Pierre? He's the...
As you want.
Yeah, so I will, just as a rhetorical introduction. So Pierre is one of the most important researchers of sleep I would say, in Europe for sure, and in the world. He's been a student of Michel Jouvet, the researcher responsible for discovering or describing paradoxical sleep in Lyon in the 60s or late 50s. And he has been pursuing the research question about what is paradoxical sleep, or also known as REM sleep. Pierre is the director of the sleep team at what we call nowadays the Neurocampus. Is a structure, is a big campus that concentrates all the neuroscience made in Lyon in one single facility. He's also the president of the European Sleep Research Society already for maybe, I don't know, 6 years already. He was vice president, secretary of the society, and he has been around the world spreading the role or the knowledge that we have about what is REM sleep, what is paradoxical sleep, is there a difference between both or not? And today he will just, he will describe some of his latest results. I just heard that his talk is very new. That's the talk that he presented in the last meetings. So it's a great pleasure to have you here, Pierre. I think it's the third time that we speak in the House Symposium. So the stage is yours. Thank you very much. And please, let's give him a round of applause. Thank you.
Muito obrigado, Claudio. Muito obrigado para convidado. Muito feliz de falar hoje. Vou falar bem português, um pouco de português, mas não demais para apresentar hoje. Talvez eu possa falar francês, se você gostou. Você gostou se eu falar francês ou não? So I will switch into English, right? So I'm very glad to be here today. Like Claudio was saying, I've been around visiting and collaborating with the center for many years. I like very much your center. I did like very much the previous house, although it was very small. And I want to congratulate here everybody and Kirsten and all the people in charge of this very nice place and outstanding place for doing neuroscience in northeast Brazil. So we were in Brasilia yesterday, and we realized how difficult for it was to do research in Northeast compared to Southeast. So I'm very glad you are here, and I wish you the best for the future. This talk, I gave this talk 2 weeks ago. It was a plenary lecture I was invited to in the French Sleep Society meeting. So it's really new stuff I will show you. Just in fact released and even not published for a lot of stuff I will show today. So I don't know if you understand this sentence, paradoxical sleep, so close and so far from wakefulness. And basically that's the idea today of my talk, whether paradoxical sleep is close to wakefulness or is very different. Paradoxical sleep means there is a paradox, right? The paradox from Michel Jouvet was that cortical activity is close to wakefulness, but then you have no muscle tone. So it's a very deep sleep. That's why it's a paradoxical sleep. So we will see if it's true or not that at the cortical level, for example, wakefulness and paradoxical sleep are close state. I invite you, by the way, just to go back, I invite you as the president to the next sleep meeting in Europe, if you wish to go to Sevilla in Spain. So that will be a great meeting we are organizing. So, of course, I always dedicate my talk to my mentor for 20 years, Michel Jouvet. So he disappeared in 2017. And then what I would like to talk about first, so this was a talk for, I would say, people knowing sleep, but not more MDs than PhD researchers. So my idea was to be simple and to be not too technical, right? So I believe that it's adapted also today because you are not sleep specialists, most of you. So the idea was to be a bit historical also, and to tell you a story more than to go in the detail of the methodology. Still, why we are studying sleep in animals? You could say you don't need animal research, and you know we have a lot of problems in Europe. Actually, they want to ban animal research. So why we need animal? If I go back to the first experiment of Michel Jouvet in the 60s, so he was a neurosurgeon. So he was able to remove here the entire forebrain of cats. So this was the idea, to find the function, to find what is in the brain responsible for function. You remove part of the brain, okay, is it there or not? That was the first idea. So that's what they did at that time. Then they make some more local lesion, you will see. But after that, we move to more, I would say, cellular and molecular approaches recently. So I would say that in 2000, we started to use this method. This was elaborated in the lab. So this is a head restraint rat, as you can see here. You have polygraphic recordings, and at the same time you go in the brain with a glass micropipette. It's a multiple channel glass micropipette, so you can do local pharmacology of drugs, and at the same time you have sleep in these animals. They sleep, amazing, huh? They sleep in this condition. So we progress a lot using this kind of methodology. At the same time, but also a bit later, we use more functional neuroanatomy. So this is also something I believe very important. You need to know where you are in the brain. So you need anatomy. Neuroanatomy is the basis of brain research. No neuroanatomy, honestly, no brain research. So you have to think about that. So here we did, I don't know, okay. Can you launch the video there on the left, please? Because I cannot launch the video. What? Ah, it was moving, okay. No, 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 it's okay. Yeah. Okay. So the method that Jouvet developed and we use also was to find a way to deprive the animal of paradoxical sleep. You see here, he has a platform, he's on a platform surrounded by water. And then at one point when he enters paradoxical sleep, there is a decrease of muscle tone here. You will see he will fall into the water because you have no muscle tone, right? So by this means, you see, he's falling. He's falling? Huh? Oh no, he didn't fall. So he has a reflex, right? Not to fall into the water. He doesn't like water. Rats, I learned that rats, they don't like the water. So it's crazy because I thought they like water. So by this means, you deprive for 72 hours, in our case, the animal of paradoxical sleep. This is the control condition, and this is the rebound condition. So here you have 50% of paradoxical sleep. Here you are 0%. It's a full inhibition. And normally you have, let's say, 10% of paradoxical sleep. Why you do that? You do that to couple this experiment with functional neuroanatomy using a marker of activated neurons named C4. C4 is a protein expressed in nuclei. It's expressed in nuclei when the neurons are activated. So it's a way to see neurons activated, and you can look at the entire brain. So this was a revolution, of course, to find out which neurons are activated during paradoxical sleep. So they do something during the state. Also, what is amazing is that you can combine this method with a lot of different markers, with retrograde tracing or any marker you like, for example, to identify the neurotransmitters of neurons. So this was very powerful. And we use it a lot. And then the last revolution is running now. Right? And this is here the use of transgenic mice. But not, you can use different type of transgenic mice. You can use pre-expressing mice in a specific population of cells. Right? That's a possibility. Here we use transgenic mice expressing Cre only when the neurons is expressing FOS. So only when a neuron is activated, the neuron is expressing Cre. How do you do that? So if you have, let's say, a condition, you stimulate the animal, but you need to inject at that time 4-hydroxytamoxifen to get the expression of the reporter gene here, which is TD tomato. The reporter gene expression will last until you perfuse the animal. It's very long. Can be 1 week, 2 months, whatever you choose. It means that compared to C4, if you make a second condition of C4, you see that, 1 week apart, C4 is expressed during 2 hours. So you have to perfuse the animal to get the C4 expression. TD tomato, you can wait. Also, you can combine the two, the red and the green. So you have the C4 labeled neurons in green and the neurons activated during the first condition in red. Same animal. You see neurons activated during 2 different conditions. First time ever in history of neuroscience that it is possible. Okay? So that was something very important for us. But not only that, this methodology goes well beyond just comparing 2 conditions. Because these neurons expressing here TD tomato, since they express Cre, you can do whatever you want with these neurons. You can play with these neurons, right? So it means that if you want to stimulate them with optogenetic or inhibit them, chemogenetic, you can do it. If you want to identify the projection, afference, or efference of these neurons, specific, you can. If you want to image the neurons using calcium imaging, you can. And if you want to identify the mRNA content of the neurons, you can. So the limitation is more people and money than really the methodology here. So what we did with this methodology, so that we started around 5 years ago with this methodology. So it's not, I'm not showing you something starting. I'm showing you something already well developed in the lab, studied like crazy by maybe 10 different people. So it's not an easy method to master. And what we started, we wanted to be sure that this methodology is working. So we made, for example, twice the same condition, like wake-wake condition. So you have the 4-HT during a wake condition. You wake 1 week, perfusion, and before that you make second time a wake condition. Just to control that TD tomato neurons are the same than the C4 stable neurons. They should be the same, right? Then the same you do with 2 different deprivation and rebound here of paradoxical sleep, 1 week apart. I don't know if you saw it was moving here because it's not moving anymore. I see the video. Because also we were obliged to develop a new methodology in mice to make a specific paradoxical sleep deprivation because the flower pot is not working in mice. It's too stressful for mice. So we have a new system, online recognition here of the state of vigilance by the software. So the software is recognizing online the state. I don't know if we can move the video. You saw the video or not? Moving? No? Okay, if there was somebody... Anyway, so this is because this is shaking the mice. The video, if you can launch the video, this is shaking. No, it's, you need to push. Or maybe you need to push on the yeah. It's not there, it should be there. Okay, it's no big deal. Okay, so the cage is moving up and down. Basically, every time the software is recognizing the state. So it means that by this mean, you have a decrease of paradoxical sleep during the deprivation, and then during the rebound, you have an increase in the quantity of paradoxical sleep. You understood, maybe I was not very clear, that why we need, and that's a pain, but we need that, we need to deprive the animal of the state because there is not enough quantity of the state to get C4 expression. So we need to deprive the animal to get the rebound, to increase the quantity of the state, to get the C4 staining. So maybe I didn't explain this. So basically, that's what we are doing. So if I go to now, I go back. So this is, I wanted to show you the first methodologies and go to the latest methodology, the trap mice. Now I go to the results. I explain to you the story with using this methodology. So if you look at Michel Jouvet's study, he removed the forebrain, you have only the brainstem here. In this cat, you see period of muscle atonia. So you have muscle atonia like this. And you have rapid eye movement too. So he thought, okay, this is the period he got, right? With muscle atonia and rapid eye movement. So he thought, okay, what is this state? So he did look at an intact cat, and he found out that during this period here, you have EEG activation. So it was corresponding to REM sleep described by the American. Not only that, it was showing that you can generate the state just if you have only the brainstem. You don't need the forebrain. Only the brainstem is necessary and sufficient. Then after that, they made lesion in the different part of the brainstem to find out which region is responsible. So they make lesion in this small nucleus here, sublaterodorsal tegmental nucleus, SLD. It has also different name, depending on the researchers. What he got? Still no video. Oh, I have a message on my mic. I don't like that. You have to do something. I don't know what it means. So, in fact, hopefully we see this one because it's important video. Okay. I'm sorry about that. I don't know what happened because this is crazy. I need to find the video. The original video. Normally I don't have any problem, but of course this time I have. Sorry. I cannot open it. See, it's here, the video is here. But it doesn't work. But I don't know. It doesn't access. Can it access it? Ah, it's here. Find it? Okay. Sorry about that. So this is the cat with a lesion of the SLD. So he's during paradoxical sleep, but he has no more muscle atonia, right? So he's moving. He's displaying different behaviors. We name this behavior, Jouvet named this behavior oniric behavior. So it means that if you remove the SLD, if you make the lesion of the SLD, then there is movement. So in a way, it is also showing that the state of paradoxical sleep is a motor state. But there is something inhibiting the motor neurons, and it's arising from the SLD. And so these SLD neurons, certainly they inhibit motor neurons during paradoxical sleep. That's the idea. So now we need to play the other video. That's a game. It's not easy, right? It's better to if I can be close to my mic. Anyway, it's okay. Can you launch it? Because he sees something. Okay, if he doesn't see something, if he doesn't play that one either, okay. Okay. You cannot launch it. Interesting. Huh? Okay, so, but I mean, that's much better to see it because it's a behavior in human during which patients, they have violent movement during paradoxical sleep. And this has been described, in fact, 20 years later than the cat of Jouvet by Mauwald and Schenk. So it means they describe the pathology 20 years after the animal model. So we had the animal model. 20 years later, they found the pathology in human. So it's a rare example. You have the animal model before the disease. So meaning that in this patient, maybe they have a lesion of SLD, and what is important in this patient, they develop Parkinson, generally, 5, 10, or 15 years later. So it's the first sign of Parkinson's disease. So everything also means that motor neurons are excited physically by the motor cortex during, we believe it's the motor cortex, because if you look at this patient, it's very elaborated, the movement, even the cat. But there is a debate whether indeed the motor cortex is driving this motor activity. Is it linked with dreaming, for example? That's a big question. So now I go with the other methodology, because you have here the SLD, but okay, we have some rain in the room. So it's the SLD, but as I told you, it's a very old method. You lesion the region, you don't know the type of neurons there. So that's where the CFOS methodology is very good. Because with the CFOS methodology, you can look at the deprivation, the rebound, and in this condition here, the neurons in the SLD driving the state should be CFOS, right? Because they are activated during this period like crazy. Indeed, in the SLD, there is a lot of CFOS-labeled neurons. We see here Brown-labeled neurons, and we needed to find the neurotransmitter content of these neurons. So we tried different ways to characterize the neurotransmitter of these neurons. So we use here the vesicular transporter of 2 of glutamate. So this transporter is only available in glutamatergic neurons. So if you have this transporter, your neuron is glutamatergic, and it is necessary to have this transporter to put glutamate in the synaptic vesicles. So without this transporter, no glutamate transmission. And like nearly all of the cells in the SLD, they express this transporter. So this was the discovery in 2011 that these neurons are glutamatergic. We also show that they are not GABAergic. They don't express the enzyme of synthesis of GABA glutamate decarboxylase. They don't express choline acetyltransferase, the enzyme of synthesis of acetylcholine. Everybody thought for more than 40 years that these neurons were cholinergic. It was clearly wrong, right? And it's still in the notebook. If you look at many notebooks, many people still say, it's still there, cholinergic, not cholinergic. Oops, video again, I'm afraid. Yeah, so what we did, but I mean, this is only neuroanatomy, right? So, great, it's working. So it's only neuroanatomy, here it's only a neuroanatomy what I show you. It doesn't prove anything. So of course you need to block the activity of the neurons to be sure that these neurons are responsible for muscle atonia during paradoxical sleep. And that's what we did here. And we have a very fancy method, genetic method, using short hairpin RNA for VEGLUT2 to inactivate only VEGLUT2 in these neurons specifically. Without VEGLUT2, no glutamatergic transmission specifically in these neurons. And you see the rats here is showing muscle activity, as you saw, during paradoxical sleep. So it's like the cat. So you remove the activity of these neurons, but don't forget, only one protein is not expressed. Neurons are there, they are alive. So we are far from the cat. And still, you have no more muscle atonia. So now just let's go a bit to the trap mice to see what it brings to us for this region, right? First of all. So this is an image I took. Yeah, could be better, but this is an image I took very recently of a mice exposed first to wake red neurons, TD tomato expressing, and 1 week later, with the rebound of paradoxical sleep, green neurons. Neurons active during the 2 states will be yellow. That's the idea. So if you look at here, the pons, right? It's the brainstem. You see the inferior colliculus. And this is the SLD region. I enlarged this region. Let's look at it a bit at higher magnification. You see here the SLD region. More green cells than red cells. Nearly no yellow cells. Different cells activated during paradoxical sleep than wake. So that's the first conclusion. Then you look here, a lot of red cells, red neurons, in the lateral parabrachial nucleus. These neurons, they are not very well known as being wake neurons. So it means that not only here we confirm here that these neurons, they are activated during paradoxical sleep, but here we have new candidates also for inducing wake, and completely different neurons than those activated during paradoxical sleep. You can see here the statistics was done by Jones Roca. You know Jones Roca, I imagine. Rocha, sorry. You know, he's in the lab actually. He's Claudio's student. He's in the lab in Lyon. I asked him to do the statistics because this is really completely new results, unpublished. I enlarge a bit the SLD. You see here the green neurons and the red neurons, different neurons. So it means that this region is very interesting, but the method shows you something nobody saw before. And we are exploring now this region with Blandine Duval and Patrice Faure, my collaborator, using the trap mice to identify in more detail the region. So now, how do you go from glutamate neurons, excitatory neurons, to motor inhibition? Doesn't work out, right? You have glutamate, excitation, so how do you inhibit motor neurons? You need something between these glutamate neurons and the motor neurons. And here, a historical paper published by Michael Chase in '91, and you see that he was recording intracellularly motor neurons during paradoxical sleep, and this is the membrane potential of these neurons. You see hyperpolarization when the cat is entering paradoxical sleep. He's inside the neurons. It's a crazy experiment, right? With a glass pipette. Then if you put a strychnine, an antagonist of glycine, you nearly remove completely hyperpolarization. I don't know if you know what is strychnine. You know what is strychnine? No, somebody know? So it's an antagonist of glycine. It's written here, or somewhere maybe. I don't know. No, it's not written. So it's glycine is a very important inhibitory neurotransmitter present at the spinal cord level. The brainstem, caudal brainstem, spinal cord level. I'm sure you know GABA, but glycine is more important there. So it's responsible. So somewhere there are glycinergic premotor neurons which are hyperpolarizing motor neurons, and they are downstream the glutamate neurons we just talked about. So now, also, let's look a bit to the new results with the trap mice. What we can do with the trap mice. You see, we found here in the medullary reticular formation, this is the inferior olivary complex, this is the cerebellum. You see, this is the rebound condition, the C4 label neurons, exactly here. If I enlarge a bit this region, this is a fantastic staining of neurons just dorsal to the inferior olivary complex, C4s during the rebound. Of course, they don't display red staining, so they are not activated during wake. First demonstration also ever. Wake neurons here are also present, meaning that here you have a wake, possibility of a wake structure, but different than the parenchymal structure. What we found out is that these neurons express glycine transporter. So we prove that these neurons are glycinergic neurons. And you don't have this kind of staining in the spinal cord. So it's not spinal cord glycinergic interneurons which are involved, it's these neurons which are glycinergic neurons, project to motor neurons, hyperpolarize motor neurons. And they receive excitation from SLD glutamatergic neurons. So we did the same that what we did for the, you need to launch the that's a pain. So we did the same that we did for the SLD neurons. We use a very fancy, doesn't play, it's right. It's a pain. So we use, anyway, you trust me. So we use a fancy method to inactivate vesicular GABA and glycine transporter in these neurons. So without this transporter, you don't have GABA glycine transmission, only in this region of the GIV here, which is the ventral gigantocellular reticular nucleus here. You see, you have no more expression of the transporter compared to control condition. If you do that, you have the same that what I show you in for the SLD. You see, you have muscle activity here during paradoxical sleep, meaning that if you remove the activity of these neurons, you don't have muscle atonia anymore. So these neurons are really those responsible for generating muscle atonia. They project to spinal motor neurons, and you get the muscle atonia. These neurons are activated by SLD glutamatergic neurons that are exciting them so that you get muscle atonia. So this demonstration, I believe, is over. Now we know. We know the muscle atonia system, I believe, and this has been confirmed by other groups after us, that indeed these two populations of cells are inducing muscle atonia. The next question is to find out what's happening in the cortex, and also whether the hypothesis is that the movement we see here are due to activation of the motor cortex. So if you remove basically the inhibition here, you see a lot of movement. And the idea is that the motor cortex is driving this movement. So we need to find out if it's true and what's happening in the cortex. But before that, I don't know. It took forever, right? How much time I have, Claudio? Because it took forever with all the problems and methodological problems. 20 minutes? Because, yeah, I was just thinking about it, because this talk took me 45 minutes 2 weeks ago, so it should, but with the problems we had, it's not smoothly going. I don't know if I go to this point now or if I skip it. I really don't know. I go for it. So the next step was to find out what is driving the activity of the SLD. SLD is driving muscle atonia, but how it works. So we try to figure out how you put this neuron on. And we found something very curious, is that in this model of head restraint rat here, if you put bicurculine on the SLD, this is what you see. You see a permanent muscle atonia during 90 minutes. So it means that if you remove GABA, because bicurculine is a GABA antagonist, so if you remove inhibition by GABA of the SLD, you get activation of the SLD permanently. So GABA inhibition is really gating the activation of SLD. Where is coming from the GABA? This was a very important question we answered to. And GABAergic input is coming from a region here in the mesencephalon, like the ventrolateral periaqueductal gray. And the DDPME, it's the deep mesencephalic reticular nucleus here. So you see these red neurons, they are neurons projecting to the SLD, and they contain the enzyme of synthesis of GABA. So they are GABAergic. It's the only source of GABA to the SLD. And what is interesting is that in this animal here, on this platform, fighting against the state, these neurons also express force. So they are very activated. So it means that certainly in this region, these neurons are inhibiting the SLD, so that is not starting to be active. We prove that. We prove that because we inactivated this region. We inactivated this region pharmacologically. I will not go into the detail because we have not a lot of time. But this has been confirmed by lesion studies. This has been confirmed by chemogenetic. This has been confirmed by optogenetic. So it's clear when you inactivate these neurons, you increase the quantity of paradoxical sleep. So it means that you need to put these neurons off to get the state of paradoxical sleep. Okay, I'm bothering you a bit now with the methodology, with the network, because how do you put these neurons off, right? Because you need to put these neurons off to get the state. And that was the next step. We did hunt the neurons responsible for inhibiting these GABAergic neurons, which are located in this region. We found out, and this was a big surprise, that the hypothalamus contained the neurons certainly inhibiting the neurons during paradoxical sleep. Why? Because the lateral hypothalamus here contains a large number of neurons projecting to these GABAergic neurons and activated during paradoxical sleep. So, contrary to what Jouvet was saying, the hypothalamus above the brainstem is controlling the brainstem to trigger the state of paradoxical sleep. And at that time, we also found that 44% of these neurons contain the neuropeptide MCH. And also, many of these neurons, they were GABAergic neurons. So that was something important, moreover, because we demonstrated that if you inject ICV MCH, you increase the quantity of paradoxical sleep like crazy. 300% increase in the quantity of paradoxical sleep, 150% increase in slow wave sleep. So this is the first, we discovered the first, and it's still the only one, hypnogenic factors, existing in the brain. This has been confirmed using optogenetic, chemogenetic, and every new methodology by many groups. So if you stimulate these neurons, you induce paradoxical sleep. So that was a seminal discovery, nearly, in fact, 20 years ago now. So now if I go with the trap mice, and this is Amarine Chancel, she's a PhD student in the lab, looking at these neurons, this region of the hypothalamus, trying to figure out, okay, what is happening? And this is where the trap mice is also very powerful. Because here you can see the MCH neurons are in blue. You see that here they express C-force. You see all the white stuff inside these neurons means they express C-force. They are activated during paradoxical sleep rebound. What is amazing is that you have red neurons also here, you see? They are different. These are wake neurons, right? Because it's a wake PS animal. So it means that this region is very complex because it's also control wake and paradoxical sleep. And that's happening a lot in the brain. So that's just a statistical analysis showing that the MCH neurons are active during paradoxical sleep and absolutely not during wake. Then what's happening during wake? So you have another population of cells, and these neurons are the hypocretin orexin neurons, famous for inducing wake. Oops. Sorry. And these are the neurons here. They are also in blue, but they are doubled, so they are blue and red. So you see they are purple. Most of the active neurons during wake, they express hypocretin, so they are hypocretin neurons. So you see the complexity, two different populations and two different type of expression. You see the enlargement. We have a little problem with the orexin, in fact, we discover, because these neurons, they are active during wake here, but they are also finally active during paradoxical sleep. So we can talk about that later, if you wish. It's a bit more complex than what we thought. Then we are hunting other populations because MCH hypocretin neurons, if you look, you still have a lot of green neurons which are not labeled by MCH. And so with Amarine, we are hunting other populations. And we found one, which is a population containing LHX6, here, protein. And you see the enlargement of these neurons here. So you can see that you have double labeled neurons here during paradoxical sleep. So meaning that we found a new population of neurons activated during paradoxical sleep. And she is still hunting other population. You see these neurons, for example, we don't know which neurotransmitter they are using. So we have still a lot to do about the identification of neurons responsible for inducing the state. So basically, this is a summary of the state of paradoxical sleep today. I just show you part of the network. I just show you SLD here, muscle atonia. But you see there are many other players. I talk also about the MCH neurons, LHX6 neurons, orexin neurons. So basically, if I want to summarize this, but I cannot go into the detail today, it's more complex, of course. But what is important as a take-out message here, take-home message, the hypothalamus is very important and is controlling the brainstem system, as you can see here. And then that's the first part of my talk. Okay, now it's the second part. I'm sorry about it. What's happening in the cortex and whether the state of paradoxical sleep is the same state than during wake, right? So this is something we can do only with the trap mice. because you need to look at the activation of neurons during wake and compare it with the activation of neurons during paradoxical sleep. So if you look, we were looking at the brainstem, hypothalamus, but now if you look at the cortex, you see immediately that it's a different story. You see here that you have a nucleus with a lot of green cells activity during paradoxical sleep. Here, mostly red, activity during wake. If you look at the percentage of cells activated during wake on paradoxical sleep, less than 10%. So mostly different systems on different localization. What is important is we found that the cortical activity overall is much more important during wake than during paradoxical sleep. That's the first message. Paradoxical sleep has nothing to do with wake, and the activation is much more important during wake than paradoxical sleep, excepting a few structures. The retrospinal cortex here, mostly paradoxical sleep, and you will see the dentate gyrus. So that's something amazing. If I look at higher magnification to the retrospinal cortex, you see here, it's amazing. The number of cells activated during paradoxical sleep is amazing in this nucleus. So it contains the largest number of C4-labeled neurons in the entire brain. And you see rarely red neurons. If you go to the visual cortex, it's the opposite. It's mostly wake here. So you have different picture for wake and paradoxical sleep. And this fits with the result we obtained with Bruna Koike, which is here, I believe, somewhere in the audience, here in the back, and Claudio. Because, of course, you can tell me that this is just neuroanatomy. So with Bruna and Claudio, we examine whether the neurons are truly more active during paradoxical sleep in the retrospinal cortex, here, compared to wake or slow wave sleep. And you can see that it's pretty clear that most of the neurons we recorded at that time were activated, more activated, and this is a big surprise, during paradoxical sleep than during wake or during slow wave sleep. Furthermore, these neurons, they were also linked with theta activity. So that's something we believe very important for understanding the function of the state of paradoxical sleep. So now, if I look at this photo, very recent, as I said, entire brain here in the scanner. So in red, you have wake, and in green, you have paradoxical sleep. You see that here? This is the dentate gyrus. I enlarge that. Okay. You see clearly a lot of green cells, statistically more than the red cells activated during wake. This is the only cortical structure more activated during paradoxical sleep than during wake. And we can use, of course, the trap mice to understand the function of the activation of these neurons during paradoxical sleep. So that's the next step, of course, of a study. How do you activate this cortical system during paradoxical sleep? That's also something we answer to. We use, again, technique of tracing, retrograde tracing, injecting retrograde tracer in the dentate, the anterior cingulate, the retrospinal cortex, finding out the neurons which are activated during paradoxical sleep, which are C4s, and projecting to this structure. We found that for the dentate gyrus, it was the supramammillary nucleus which contained neurons activated during paradoxical sleep and projecting to the dentate. Only this very small nucleus here in the hypothalamus. So he's in charge of activating the dentate. For the neocortical structure, it was the claustrum. Nobody knows. Nobody knew the claustrum at the time when we discovered that. Now there are more papers on the claustrum. So the claustrum is the only area projecting to these two structures with neurons activating during paradoxical sleep. If I look at the claustrum, if I look again at the trap mice, that's very interesting also to look to the mice, to compare wake. You see the red neurons here, to compare wake activity in the anterior cingulate cortex or the claustrum, right? You see anterior cingulate cortex, mostly red neurons, mostly wake structure. Some green neurons here, but of course, much less activated during paradoxical sleep. Now, if I look at the claustrum, that's also something quite crazy we get with the trap mice. You see different population of neurons. The red neurons activated during wake, certainly playing a role in activating the cortex during wake, and the green neurons activated during paradoxical sleep, different neurons. So you see the trap mice is showing you again for the first time, demonstrating that it's not the same neurons we are talking about. So this is, I have two more slides. So the question, of course, we are now trying to answer to is, what is the function of the activation of this limbic cortical structure during paradoxical sleep? We believe that if we find why this very specific set of cortical structure is activated during paradoxical sleep by the claustrum and the supramammillary nucleus, we find that we finally understand the function of paradoxical sleep we have been looking for more than 50 years. One hypothesis, of course, is, and I'm pretty sure about that, is that this structure induces dreaming. In particular, dreaming with motor activation. Right? So it's like you store scenario in these structures, and these structures are all important for memory. You can then generate a scenario with some motor activation. You can even test new behavioral strategy, motor strategy. You have no problem. You are not moving. You have muscle atonia generated by this system. So you can try a new motor strategy, right, in your bed. If you have not a problem like the patient I was showing to you. Another possibility we are trying to address is whether it's important for mood regulation. Paradoxical sleep is very well known for having a role in mood regulation. So we have a model of depressed mice here. It's a model of chronic stress, light chronic stress. And then what is known already is that in this case, after several weeks, you have a 30% increase of paradoxical sleep in this model. So we are looking at the activity of this system here in this model to see if it's modified. Because if it's modified, then we will be able, with the trap mice, to reverse maybe the phenotype of depression. And this might be a step to understand depression. A last slide about something very strong, actually, is the link with paradoxical sleep and memory. In fact, around 2000, people started to think that the state of memory is non-REM, slow wave sleep, and paradoxical sleep, is not that important. Before 2000, paradoxical sleep was the only state involved in memory consolidation. But you see this number of papers here in very good journals published since 2016. And I summarize here what they found. And in fact, it fits quite well with our results. And in fact, one of the paper is published in collaboration here with the center from Daniel Almeida Filho, based on the data we also acquired together. So basically, the idea is that here you have memory consolidation, and that the pathway from the lateral supramammillary nucleus to the dentate on CA2 is important for memory consolidation, here spatial memory, and here social memory consolidation. That's a new paper. And we show also that the retrospinal projection to the dentate is important. It has been further shown that here newborn neurons, 4-week-old adult-born neurons, are important and are activated during paradoxical sleep and consolidate memory. Then you have another here population, the MCH neurons I talk about. And these neurons, they project to CA1, and it has been shown that they play a role also in memory consolidation. But what is strange is that there is a paper showing that maybe it's important, the activation of these neurons, for forgetting. So that's a bit strange. And then you have a very recent paper by our friend in Bern, showing that something very strange, because as I show you that there is not much activity in the prefrontal cortex during paradoxical sleep. And they found that indeed the cell body are inhibited by parvalbumin interneurons during paradoxical sleep. But they reported that the dendrites are activated by a series of interneurons here. So you have activation only of the dendrites in the prefrontal cortex, not the cell bodies. We don't see that with C4s, obviously. And they show that this is also important for memory consolidation. So you see a lot of exciting stuff, and I'm sorry to have been long and with all the problems with my talk, but I ended up here and showing you the people in charge of doing all the experiment I've been talking about today. Thank you.
Thank you very much, Pierre. Great talk. It's great to see that you went down to the spinal cord and up to the cortical structures as well. So I just want to know, does anyone have questions to Pierre? Anyone? You have one?
As usual. If nobody does, I'll do. Hi, Pierre. Thank you for the talk. Great. I'm sorry, I think I missed the first part. So if my question is you already explained, please ignore me. I was wondering about your trap mice, right? So your trap mice, do you detect through Cree-dependent activation, correct?
In fact, it's force-dependent activation, okay. Right? If they express force, they express Cree, and they express the TD tomato.
Okay.
Yeah.
So are all the cells capable of doing this recombination equally? I mean, I was just wondering whether it's inactivation, whether if you can detect them all.
So that's the good question. That's a very good question, and that's why we did control experiment with the same condition twice to compare force and TD tomato expression. I would say that we have an intense debate in my lab about this. Some people are not that happy with the methodology, saying that, okay, there are some regions in which you don't have TD tomato neurons or less TD tomato neurons that C4 label neurons. So there is a debate, but I'm not negative. I'm very positive about it. I would say that, for example, in CA1, it's the unique region in which it's not labeling a lot of neurons. So we don't know why. Here it's clear, but for the other region in general, it's and in fact, Claudio, you did an analysis with me to correlate the number of TD tomato with C4s in many structure, right? In the wake condition, there is a very good correlation. So I would say that it's not exactly the same, of course, because it's a very complex transgenic mice. And you need to inject tamoxifen. So it's very complex and delicate methodology. So you cannot say it's exactly the same than C4s. No. But, I mean, it's working. I mean, I like it. Of tamoxifen, yeah. Yeah. So just to tell you, so the methodology I've been showing to you here, we needed 5 years to improve it progressively. So we did a lot of modifications of the protocol. At the beginning, we started with to inject IP for HT, and we increased the dose, we decreased the dose, so we tried different concentration. So the higher was the dose, the better the staining was. But then you started to have a side effect, which is a problem. And then finally we put some tamoxifen into gelatin. So we give this gelatin to the mice now. They eat it very quickly. And so you have no more side effect. And so that's the latest progress in the methodology we have. We also found, I believe, the right dose. And you have to know also for wake, it's easy, because for wake, you just, you have 100% of wake. You see paradoxical sleep. If you wake up the animal by injecting IP, you have a problem. You don't have only paradoxical sleep, right? So it depends also on what you are looking at. I would say for wake, for example, it's perfect. For paradoxical sleep, that's why I've been fighting a lot also to improve the methodology, because that's a pain to study. I like paradoxical sleep so much, but that's a pain to study this state, because you have less than 10% of paradoxical sleep. So it's very difficult to study a state, a rare state.
Kerstin?
Yes. Yeah. Yeah, that was something I wanted to try to label with C4s. Also, you can use the trap mice. Animal P at P7, P8, P9, P10, this, because that's normally I expected to have 70% of paradoxical sleep at this age, 70%. So I thought, okay, if you do a perfusion at that time, you should see the network. Although the network is not completely ready, so the cortex is not ready, for example. So I didn't expect to see the cortex, but at least the brainstem. But I mean, this is something we tried. We didn't complete yet, and it was not very successful. Although C4 is working. Something important maybe for you, if you want to use the trap mice, is that at one point during the lockdown we injected a mice with 4-HT. And then there was a lockdown, we didn't go in the animal facility for 2 months. So we went back there, perfused the animal after 2 months, and it was perfect, the TD tomato. So it means that I imagine that if you want to study the development, for example, to inject a newborn mice, and then to wait until the mice is adult and to make a C4 staining, it's possible. So and this can be something.
Other questions? I have one question, Pierre. It's nice that you use neuroanatomy to describe function, right? So most of your talk is describing, you take a picture, neuroanatomical picture, and you describe function. But we know that during REM sleep, it's not only muscle atonia. You have muscle atonia dominating the state, but we also have twitches, muscle twitches. Yeah. So my question is whether you think these muscle twitches are locally generated in the spinal cord because the inhibition of the motor neurons are not, something goes wrong, or is there any structure that is controlling the pathway of the ventral gigantocellular nuclei to the spinal cord to abolish the inhibition of these motor neurons so twitches can be present? So any comment about what's the role of twitches during REM sleep?
So the twitch is, we know a bit how it works, right? In terms of the mechanism, at least at the motor neuron level, right? So we know that you have glycine GABA inhibition, so that, I mean, inducing muscle atonia, and we know that you have from time to time some movement. Still, even if you have a strong inhibition, you have this small movement. If you put a glutamate antagonist, they are gone. So we know it's glutamate input. So it means that, as I said, at the same time you inhibit, and you inhibit not for nothing, you inhibit because you have this glutamate input. And from time to time, if there is a strong glutamate input, too strong glutamate input, it override the glycine GABA inhibition. The question you are asking is where it's coming from. So there is a debate, and it depends also maybe in the newborn versus adult. In the newborn, the cortex cannot generate these twitches, and there is one researcher in the world saying that these twitches are key for setting up the sensory motor system. So it means that you need paradoxical sleep, and you have 70% of paradoxical sleep in the newborn to set up your sensory motor system. That's the idea. And these twitches are important because when the animal twitch, there is a sensory feedback to the cortex, and this sensory feedback helps to set up all the sensory motor system. So that's the idea. But it cannot be generated by the cortex, so there is a brainstem generator in the newborn. What we believe is that in the adult, you still have, of course, this brainstem generator, but we believe the cortex is playing a role also to generate the movement. So it's controlling the brainstem generator. So it's not only at the level of the brainstem. So that's the idea, but it's not proven yet.
Well, maybe Monique.
Pierre, you know this paper, the recent papers from Keane and Chang et al. In 2022 that are showing that the neurons from the lateral supramammillary nucleus can transmit the novelty signal to dentate granule cells during by shaping the engagement of the dentate during encoding of spatial memory. There is also this paper that is showing that the neurons of the semlateral are also important for consolidation during REM sleep. Mm-hmm. And you are showing with the trap mice actually that the neurons that are activating the dentate, and there is only a few neurons activated during waking, isn't it?
During what? Wake?
Yeah, awake, when the encoding of new events can happen. And the neurons that you see occur during REM sleep, and they are only C4s, positive. So this is not, it looks like it's not the same population of granule cells that will be involved in encoding memory, spatial memory in the dentate, and be involved in the consolidation of this memory during REM sleep. How can we reconcile that?
But that's an important question. Thanks a lot, Monique. We are addressing actually with Claudio, because we have recording of the dentate actually. So we are trying to complete the paper about with recording of neurons there. And from what we see, the activation during paradoxical sleep of neurons is very rare, right? And you don't see activation of the same neurons across different multiple paradoxical sleep episodes. So it's like it's like if during a given paradoxical sleep episode or a given paradoxical sleep period, it's like you activate a setup of memory engram, a network, and then maybe a bit later, a different memory engram network or group, and so on and so on. And during wake also, before, it's a different memory engram system which is activated. So it's very interesting to see that you never activate the same neurons twice. If you have wake, paradoxical sleep, paradoxical sleep, paradoxical sleep, never. So it means it's really activating different set of neurons, and certainly I would say it doesn't prove, but we believe that it's showing that you activate different memories every time. That's the idea.
Hi, I'm not very much inserted into this area. I'm from computer science, so it's a generic question. But how would this work apply to human brains? How similar is mice brains to human brains?
That's a crucial question. Huh? Of course, in human, it's very difficult to look at the activity locally. Of course, at the cellular level, molecular level, that's why we are using animals. But like you see the patient, RBD patient, so there are, for example, data showing that this patient, he has neurodegeneration of the neurons I was talking about in the SLD and JV. Right? So in this region, you have neurodegeneration. If you talk about dreaming, for example, the retrospinal cortex stuff, I'm trying to find the region corresponding to the retrospinal cortex in human. Because, I mean, if we are able to find this region in human, we can study and maybe link the activity of this region with dreaming in human. Up to now, I didn't find, because there is a retrospinal cortex in human. It's a small piece of tissue. I don't believe this is corresponding to a retrospinal cortex we see in rodents. So it's not exactly, so the atlas in human is not showing the same, I would say, than in rats and mice. But so we need to walk, maybe I was thinking of monkeys, to go to monkeys to find out, but I need a monkey brain. So if you have a brain here, it's a call for you. If you have a monkey brain, right? And if you could perfuse it for me during, just after a long period of paradoxical sleep, and then I would look at the CFOs, it would be really very important for us. And I believe it's feasible, right? Because you need, it's very consolidated sleep in monkeys, not like in rodents. It's more like human. So they have long period of paradoxical sleep. So we could perfuse one animal 1 hour after this period. Of course, you need to record a bit EEG and EMG. 1 hour, and then we would see the network in monkeys. And so it's much closer to human.
Yeah, I see. Thank you.
So I think we are ahead of time. So thank you very much, Pierre, for the nice talk, and thanks for the discussion.
Okay, so after Professor Pierre's very interesting talk, we're going to continue our event schedule with our second Flash Talk session. So just like we did yesterday, the poster presenters were asked to submit a slide summarizing their work in a single slide, and they will talk about what they're presenting in their poster in 60 seconds. So it's very easy feat, right? That's why they're not nervous at all. But yeah, let's start by Ana Augusta, which will be the first one presenting.
Hello, can you hear me? Yeah. Hello, everyone. It is great to be part of House Symposium 2023. And thank you to the organizers for this session. My name is Ana Augusta, and I work as undergrad student at Neural Network and Epilepsy Lab, led by Professor Claudio Queiroz. At the moment, the lab focuses on the role of different phytocannabinoids in neurohyperceptibility. You may know there are many studies investigating the pro and anti-seizure effects of cannabidiol, CBD, and THC. However, let's know about the other phytocannabinoids. Just a second. If you want to know more about the CBG, cannabidiol, and cannabinol, CBN, we invite you to visit our post. We have some intriguing results and love your opinion about it. Thank you.
Thank you, Anna. And next on, we have another Anna Campos.
So, hi, good morning. My name is Ana Clara. I am an undergraduate student here. I'm a biology major, and I'm part of Ethogenesis Lab under the orientation of Professor Daniel Takahashi. So, I'm going to talk a little bit about my poster, which is called Infant Development in the Common Marmoset. So, among the primates, only us humans and Callitrichidae, which is the group marmosets are a part of, are known to have cooperative reproduction and also vocal turn-taking. And because we share those characteristics, it means we can study marmoset behavior development and behavior in general, and also compare it to human behavior. And it can provide us with a comparative approach to understanding human behavior. So even though we have very extensive literature on human behavioral development, there is a scarcity of that kind of research in marmosets. So here we're trying to address that gap in the literature by asking first two very basic questions, which is, do social behaviors develop? Can we observe that development in marmosets? And how do they change? How can these behaviors change over time? And secondly, is the development of different behaviors co-modulated? Can we see different behaviors, maybe interacting with each other's, with other behaviors during development? So those are very basic questions, and we observed the development of 4 captive marmoset infants during 5 months, and I think we have some interesting results. And if you want to know more about it, you can visit me by my poster. Thank you.
Thank you, Anna. And next one, we have José Firmino.
So, in contrast with human vocal development, the people believed that primates undergo 2 little changes during the vocal development, with no role in the vocal parent's feedback. However, this knowledge has changed with Takahashi and your group when they showed that marmoset monkey, a New World primate, have your vocal development guided or directed by vocal parent feedback. But this evidence was just to one context. In this work, I can show to you that marmoset monkeys, which pass by playback experiments, would learn to vocalize short contact calls in a switch context. And in other words, in a context outside the original context. These results show that marmoset vocal flexibility is larger than previous thought. If you want to know more about my work, check my banner.
Thank you, Firmino. Now, please welcome Juliana Avila.
Hello, everyone. My name is Juliana. I'm a PhD student here at ICE. And my poster is called Mental Health, Sleep, and Psychosocial Safety Climate in Higher Education. These are some of my thesis results, and the ones that I choose to bring for you today are about mainly there's a lot of dreams, nightmares, and environmental risk for development of mental illness and sleep disorders. So if you're curious about it, come check it out. I'm right next to the coffee break table. We'll talk.
Thanks, Juliana. And the next one will be Lucas Lira.
Good morning, everyone. So first, I have to say I was scheduled to be a talker here, but luckily for all of us, Professor Sergio will come join us later this morning to talk about this work and the work in our lab. So previous work shows that we find that there is a big difference with the different protocols of anesthesia. So we find that a wake cat have an entrainment in the brain that follow the frequency in the stimulus that we show. So we try to find how to see if the size of the location, what the stimulus change this entrainment. So in the right, we find that very recent, that is not about the size, but more about the continuity. We find that little stimulus causes no entrainment, and that stimulus with the middle part missing, there is no entrainment, but a bigger one have entrainment. So this is reveals a perceptual binding in the natural geniculate nucleus, and we will be looking more deep in next year about this topic. So thank you.
Thanks, Lucas. The next one will be Pedro Varela.
Hi, my name is Pedro Varela, and I'm here to talk about a little about artificial neural network model for marmoset call classification. So marmosets demonstrate vocal flexibility, indicating convergent evolution. So studying intervocal communication adds in understanding human speech evolution too. So, however, this vocal communication necessitates manual classification, which requires time and expertise due to the variety and complexity of marmoset calls. So, this study presents an automated system using a hybrid convolutional recurrent neural network called TweetNet, achieving 95.84% in controlled settings and 86.21% accuracy and field data. It maintains high accuracy across different marmoset calls and requires minimal training data, approximately 35 minutes of video. So this robust model can generalize classification, as we can see, and facilitates the large-scale experimental data analysis. So if you want to know more about this model, come check out my banner.
Thanks, Pedro. And now please welcome Ricardo Carvalho.
Good morning. And for starting this conversation, I need to talk about acoustic niche. This term exemplify how a vocal signal evolve via ecological feedbacks, and this evolution makes a soundscape complexity you can see in the environment today. So I studied the the acoustical niche of marmoset for make that recording 2 wide groups of marmoset for 3 months. And the end of the recording, I see the vocalization with spectrograms and detect marmoset vocalization and vocalization of the other species. In my presentation, I show use the marmoset acoustic, which animal presents in this niche, and what their strategy. So that's it. Thank you. Bora Bahia.
Thanks, Ricardo. And for our next and last speaker, please welcome Thaís Lima.
Oi, pessoal. Eu vou falar sobre o meu trabalho sobre meu nome é Thaís. Eu faço parte do laboratório de visão aqui do Instituto do Cérebro. O meu trabalho é falar sobre estudar sobre o efeito dos estímulos, da excentricidade dos estímulos na frequência gama em VIII de macacos caputinhos. Então, muito se fala sobre a atividade gama e sua importância no processamento visual e de como a força de acoplamento e a probabilidade de sincronização gama em redes neuronais pode facilitar processos como ligação perceptual. Sobre essa frequência, sabe-se que ela é fortemente dependente de estímulos, como propriedades específicas do estímulo, como tamanho ou velocidade, e o nosso objetivo é mostrar a variabilidade da frequência gama de acordo com a excentricidade no campo, no córtex visual primário, onde podemos ver que em regiões mais centrais de representação foveal, a frequência gama ela é maior quando comparada à excentricidade periférica, mostrando assim, enfatizando assim a importância significativa da frequência gama na dinâmica, a importância significativa da excentricidade do campos receptores no córtex visual primário na dinâmica da frequência gama. Obrigada.
So thanks, Thais, and with that we close our Flash Talk session. I want to thank to congratulate again everyone who agreed to participate. It takes some courage, and you all did great. So now we're going to leave the auditorium for our first coffee break of the day, and you have the opportunity to visit all their posters and the posters from the people that did not have Flash Talks today. But don't worry, you don't need to hurry up and ask them to do it in 1 minute again, because we're going to have a second poster presentation at the afternoon as well. So if you don't get to see all the posters at the morning, you can go back to them in the afternoon.
Let's make this coffee break a 20-minute, and then we'll meet at 10 after 11. All right? Thank you. Isso foi bem enrolado. E também o pessoal chegou tarde, 9 horas de tarde. A gente tem que marcar o... Tá desligado. Tá desligado. Welcome back. So let's go for this next session.
Hello.
We'll have two talks in this session, Professor Sérgio Neuschwander and Ana Luisa. So, Sérgio, please go ahead. The stage is yours.
Many thanks. I'm very happy here to be once more in our house symposium, and I hope that I have a completely new slide. So my goal is to show again the slide Lucas has shown with a little bit of more context. Visual entrainment. First, I would like to show this wonderful painting of Leonardo da Vinci. It's called O Salvador do Mundo. And this painting reveals something very special in the technique of Da Vinci, that is hindering the eyes. This is something unbelievable, as everybody knows, in the Mona Lisa, the Gioconda. In the words of Juan Arias, I thought I have translated this, so I'm sorry, I will read in Portuguese, that appeared in El País. He describes, Leonardo tinha nervos em seus olhos e cérebro que nos faltam, ou conseguia isso com a força de sua curiosidade inata e infinita. O que podemos concluir é que, se o cinema existisse nos tempos do gênio, ele teria sido capaz de observar cada fotograma separado do filme. And why this colocação de Juan Arias is very interesting, because many argue that Da Vinci was capable of seeing very fast, like single frames in a film. And there is, in fact, a whole academic discussion about this, mainly because Da Vinci describes that the dragonflies use the four wings, but in opposition. Now, it's only possible to verify this if you have a very slow, high-tech camera like this, that you can do a very, very slow motion, so you can really appreciate the very complex flight of the dragonfly. But Da Vinci was aware of it. So that's why this notion that he was capable of seeing frames with a kind of a superpower, high temporal resolution vision that normal human beings were not able. And in fact, he was, of course, very impressed, and he has some thoughts about vision. Very nice to see this gravura, because you see, it was not clear for him where to put the observer, the eye in the star. Now, of course, Da Vinci was not aware of the whole complexity of the retina, and here I show a wonderful picture that is, in fact, an imaging of photoreceptors of a human retina. These are the different photoreceptors that are false color. That is very similar, and you know this, like a CMOS camera, a digital camera. And in fact, this very idea that you have a mosaic of photoreceptors that in fact would be like the single pixels of a camera holds. In the same way the camera has a reading of a small collection of pixels and do some computation on this, the retina does the same. So this is a wonderful photograph. So this is real data. Now it's important always to tell if this is real or not. So this was a different staining of a single cross-section of the retina of a mouse. And you see here the bipolar cells, wonderful, the amacrine cells, the huge inner plexiform layer that connects all this activity, different from a CMOS camera, and the output that is carried out by the retinal ganglion cells. Now, what happens if you see a film like this in an old television? What you see? A wonderful film that makes sense. What you see is an integrated motion over frames, that in fact made possible the whole cinema technology. So Etienne Marret explored this, and he started playing with frames, and he understood, like Edison, that in fact the visual system integrates across frames in a way that you can then have fluid motion representation. This all happens in the retina. But there are other things that happens in the retina when you see a television. And this is something that is unexpected because nobody ever think about this. And what happens is that the retina follows the flickering of the television, a mass action in the retina of many of its elements. Many different types of retinal ganglion cells are capable of following these external rhythms. So the question, an interesting question, how this is possible? Knowing that, a paper of 1980 of Lemucle, knowing that the retinal ganglion cells, both the X and the Y types, has a clear cutoff for very high temporal frequencies. But I know the television and many of our vision displays use scanning that are much higher than this. And I assure you that the retina follows it, although you are not seeing it. What happens in the retina? We have made a series of experiments in our laboratory using away cats. In fact, the protocol allowed us to record from away cats and then deliver different types of anesthetics and study the LGN that then can be considered a proxy of the retinal activity. The results, I understand that it's difficult to make sense of all these diagrams, but these stripes means that you have very strong peaks in the sliding spectrum here, or here's the sliding correlation. Here are the response of the cells in these panels. And there is a clear difference for the awake and cats that were anesthetized with halothane. If you show a monitor that flickers, the retina can follow this flickering, external flickering of the monitor. As you see, it was made at 100 hertz, and the oscillation of this retinal ganglion cell happens to be exactly at 100 hertz. It makes sense. If the retina, or this particular cell, is following the screen. Now, if you use a source of light that doesn't contain any flickering, it's completely flat, like a LED. In case of the awake cat, there is no component, oscillatory component, but there is, and this is very interesting, one that we knew happens to occur in the retina, the famous gamma oscillation. And I will explain a little bit more later. So here, a comparison between awake and the cat under halothane. You see clearly if there is no flickering input, there is no oscillation in the retina if the cat is awake. However, under halothane, always you see, independent of the quality of the characteristics, temporal characteristics of the stimulus, you always see an oscillation. So this oscillation in the retina, the gamma fast oscillation in the retina, is generated internally and doesn't happen for the awake cat, only if in the awake cat the input is flickering. So, conclusion here, this data clearly shows a mass action of these highly oscillatory inputs. The retina is capable of following these fast rhythms. 100 hertz is very fast even for the retina. Now, this is a wonderful summary of these experiments, because here I use the monitor at different frequencies. We tested up to 120 hertz. And you see that there is a lot of oscillation happening exactly at the same frequency of the monitor for the different frequency studies. studied. 60, 75, 85, or 82, 100, and 120. Some examples, very strong coherence, of course, because all these signals are phase-locked to the oscillatory input. There is no difference between two analysis windows placed in the beginning and at the end of the process because the frequency doesn't change. But why the frequency doesn't change? Because the monitor is always at the same frequency. Now, see what happens when we deliver halothane to the cat. Now, independent of the oscillatory frequency of the monitor, you have a merging of all these dots, meaning that this frequency that is now generated internally in the retina follows a different, if you want, crazy rhythm that has nothing to do with the oscillatory input. So the halothane is capable of very deeply, very profoundly changing the neuronal dynamics in the anesthetized cat. This is summarized with this plot, and I will invite you to read our paper in the Journal of Neuroscience that we published this year. I'm sorry, because I have forgotten to put our reference here. Now, why this is interesting? This is interesting not because of gamma, for sure, but because entrainment tells you about the temporal structure of the responses. The entrainment tells you about synchronization, of course not about oscillation, because the oscillation is extrinsic. And there are many reasons for this huge interest on synchronization. One goes back to this wonderful paper published in 1896 by the group of VIII, Alonso and Martin Usray. José Manuel Alonso. You see, I will read the phrase, moreover, these neurons overprojected to a common cortical target neuron, where synchronous spikes are more effective in evoking a polysynaptic response. The message, if you have synchronous spikes, the cortical cell has a higher likelihood to fire. This increases efficiency in the transmission of the signal from the LGN to the cortex, meaning that you have better efficiency, meaning that you have a better encoding of the stimulus. This is the whole idea of why synchronization is important. This appears in a series of, I just took one, works of Marcus Meister when he was in Harvard. Here a nice example showing that concert spikes encoded the information in the retina of the salamandra. Very, very nice work. But also recently, there was a huge discussion about the meaning of this entrainment of signals by the LGN. Here, particularly a recording from the mouse in our awake mouse. And you see very strong entrained signals, very different when you deliver isoflurane, where there is a small decay and the frequencies is completely different. Exactly as you have shown, we have shown in the cat. Although these people do not cite our paper or our abstracts at that time in this context, it cites in another that I will show. It insisted that these oscillatory signals are important for binding. Although what they are showing, and I cannot really go into detail, what they are showing is a very strong phase-locked signal that is in fact generated by an external oscillatory signal. Now, there are many examples of phase lock in the retina. This was known, but nobody was paying attention on exactly this problem. Here are examples of these, one of these José Manuel Alonso paper. They are mapping receptive fields and describing the oscillatory dynamics, the synchronization dynamics in the retina. They are not describing oscillation using a different type of anesthesia than what you use, different from halothane. But you see many examples of a very strong phase-locked signals, showing that the retina, in fact, and this was known, is very capable of following very fast rhythms. Now, why this whole thing is interesting And this is because, here an example, because there are other papers showing, this is a recording from the retina under the microscope. So it's a flat mount retina of the mouse. Could characterize the cell types very nicely because Roy and the lab of Bloomfield in New York, they can feel the cells. And so they know where they are recording from. And you see some predictions we made with paper in the cat, that if you have continuity of the stimulus, you have a very strong oscillations. This is in vitro, but it's a different animal than a cat. So it brings an idea that this may be a universal mechanism in the retina. Moreover, they have tested behaviorally, and they could block the communication of these retinal ganglion cells because, of course, this synchronization of oscillatory signals in the retina depends on a connectome. And this connectome is provided by these particular cell types that are the polyaxonal amacrine cells. As I have shown to you in the beginning, in the plexiform layer, they connect horizontally very distant retinal ganglion cells. This must be important for something. And they thought this is important for behavior because the mouse that has these polyaxonal amacrine cells blocked, they lose the capability of discriminating object, exactly as we predicted with our work in the cat. This was our suggestion, a very interesting paper published in Nature Neuroscience, although without follow-up, showing that the frog lose the ability to jump if you block this horizontal connectivity in the retina. So the whole idea here is that the entrainment exists. This is a very strong, very clear phenomenon in the retina. Both the oscillations generated intrinsically and the entrainment of responses are dependent on stimulus properties, such as size and contrast. This has been shown in other papers, even in the monkey. Peak frequencies for intrinsic and entrained rhythms overlap. Retinal oscillation are strongly phase-locked to stimulus onset. So it suggests that, in fact, maybe the entrainment reveals the synchronization dynamics in the retina. And this is exactly what we thought. And we had some predictions based on our previous study, showing exactly what you have seen for the isolated retina of the mouse. In the cat in 96, we demonstrated that depends on the stimulus continuity, size, and contrast. So we could think maybe the entrainment follows exactly the same basic, if you want to say, Gestalt rules, reasons for binding, not with gamma, but now with entrainment. And this was the project I suggested to Lucas. Where is Lucas? Lucas is there. So we are doing some experiments, and we could exactly demonstrate this. And of course, we are happy because this opens a new avenue of thoughts in relation of the synchronization phenomena in the retina of the cat. If you use just one stimulus that is very small and restricted to the receptive field of these very ganglion cells, that could be an X and Y cell, if you flicker it at 100 hertz, the cell responds vigorously, but it doesn't oscillate. That's why you see no coherence, you see no phase locking value, you see a dispersion of phase. The cell doesn't oscillate, although respond. Now, remember that this can be understood taking this into account. Of course, these cells do not follow flickering stimuli. They have a cutoff at 1632, how they will work at 100 hertz, 120 hertz, 60 hertz. They will not follow this. So it makes sense if you're restricted to the receptive field of the cell. However, if you stimulate everything, the cell follows. This is what is very nice. Meaning that you have exactly the same continuity rule here, bridging the periphery and the center. Also a condition that there is no synchronization, no, better to say, entrainment, no phase locking, although the flickering is there for the cell always, you see, in the center. This behavior appears only if you have a continuous large stimulus, exactly what we have described and published in Nature many years ago, what the group of Roy and Bloomfield published in Neuron. So the message is, we propose, like the retinal gamma oscillations, response entrainment requires active synchronization. This is something new that we can really test. And does not reflect a phase locking that is passive. It's like saying the following. The retina uses this enormous machinery capable of affording, if you want to say, synchronization horizontally to encode better or more efficiently the stimulus. Perhaps it makes sense. Remembering, thinking that a readout mechanism, from the viewpoint of a readout mechanism, doesn't matter if the retina follows external rhythms or generates its rhythms internally. We argue that visual processing is robust against the influence of such fast tonic oscillatory inputs. This, of course, I would say, comes from our common experience. You see all these devices that are flickering all the time. If you go here and see this monitor, you have a lot of artifacts when you move the eyes, because in fact, there is a very strong oscillatory signals in all these devices. You do not see it. This is important or not. Maybe it reveals something, as I try to show to you, some basic mechanism of operation of the retina that use synchronization to bind things, but not oscillation, because oscillation is not there when the animal is awake. Recently, the group of Martin Fink and the Anstrugman Institute has shown, this was a kind of a very nice and sophisticated paper, but also something that I was not really expecting. He shows that the propagation of these entrained responses are limited in the cortex. And he argues that this is the case because of the band-pass characteristics of the cells. Perhaps this is the most important figure. You see the flicker signal in the retina propagates to the LGN, and really you can consider the LGN a proxy of the retina, at least in relation to this, but it doesn't propagate to VIII, other cells of VIII, sorry, laminate 2-3, propagates to the neurons that are receiving inputs from the retina, this is important, but it doesn't propagate to other layers in the connectome of the cortex, and doesn't propagate to the hippocampus. This is very important. Why this is important? Because recently the group of VIII proposed, and in fact I think that they really developed the whole story on this based on these entrained signals at the gamma band. So I put a small video because it's better that she says it for us.
Appears crucial for maintaining the brain's defenses against Alzheimer's disease pathology. In two new studies in cell and neuron, by exposing mice to sound and light that is crucial for their frequency, Psi-Lab scientists led by graduate student Anthony Martorell and Picower fellow Chiniko Rupanadakan enhanced the power of gamma rhythms in the brain. This produced marked reductions in the tau and amyloid beta protein buildups that are key pathological features of Alzheimer's. This sensory stimulation significantly improved the memory of mice engineered to have Alzheimer's disease compared to unstimulated controls.
This is a bold finding. You show that the mice exposed to flickering stimuli And we know, of course, now you know also that the retina is capable of following these gamma rhythms that are generated externally. They are capable of improving their Alzheimer condition. And that this was a surprise. And of course, this made a huge furor in the community because this could be potentially an interesting method for improving the condition of Alzheimer patients. That is really true? Let's find out. And I'm very glad to collaborate with Kerstin Schmidt exactly on this, eventually. And we bring our basic science in the cat to a clinical context in the Alzheimer's disease. Many thanks.
Thank you, Sergio. Beautiful. So we have time for maybe 2 questions. Anyone?
Claudio, please.
No, very nice talk, Sergio. It's great to see
Criticism.
No. Come on. I'm happy to see how this story evolved in time. So congratulations for you and your group. It's really nice. My question is whether you have any ecological function for this flickering, for this following flickering activity, because in nature, I would say that you do not expect a flickering at all, right?
Wonderful question. There is no ecology possible to explain this. And this is exactly my point. The flickering perceived reveals a process that exists in the retina that has nothing to do with oscillation, has to do with synchronization. And now we can test this. Now think how it could happen. When you move your eyes, you always fall into different, not always, but many times, to completely different scenes. Perhaps these very synchronous first volley spikes in the cortex improve efficiency exactly at this very time window. And this is important for sending it very fast to the infratemporal cortex. Predictions, and I had one. What's the most important object you can think for a human being? A face. You can consider a face as a visual object that is very important ecologically. Now, you could divide the face in pieces and present it with different offsets in time and test if you have the synchronous one being important for discrimination. Why? Because face discrimination happens extremely fast, something that Simon Thorpe has shown to us, even when he was in Paris many years ago. And this is something that we do not understand how this is possible. Maybe the retina contributes to this. If you go and you see in a paper published in Journal of Neurophysiology of Pedro Maldonado, you find that the coincident spikes happens very fast, very early in the cortical response. Maybe this comes from the retina.
More questions?
Essa é uma jogada ensaiada, viu, gente? Passou a noite toda ensaiando.
So first question, Leonardo da Vinci, does he say that he maybe used peripheral vision to see the movements of the dragonfly? Did you find this somewhere?
Yeah. In case of the dragonfly, this will be, even the peripheral vision will be very difficult. However, I have a response. Of course, I have a response. And this, nobody knows if this is correct or not, and I have been discussing this possibility with some experts, although it's not conclusive. If you take the wheel of your bike and turn the wheel of your bike, can you see the raios? How you say this, Tarcísio? Os raios? The spokes. Can you see the spokes? No, you cannot see, because they have such a fast angular velocity that, of course, the cells cannot follow this. However, if you move your eyes and you see for a brief 100 milliseconds, even less. Why? Because I think there is a coincidence of the angular motion of the eyes and this, because if you do it in the other direction, it will not work. Eventually, Da Vinci was aware of it and was doing this with the dragonflies.
I think so.
Maybe. I find unlikely. Yes.
It's moving its wings and actually moves. So a good observer would probably do that and interpret as the motion of the dragon.
It's a wonderful response. So you make the dragonfly slower because of temperature. Very nice answer. Now I ask to you, academic, do you think that you can explain these wonderful paintings by science? No. Thank you.
Tudo certo?
Legal. Vou botar o som.
So thank you, Sergio. And for our next speaker, I have the pleasure to introduce Ana Luisa, who is currently a PhD student here at the Institute. She's from the Computational Neurophysiology Laboratory, so my lab colleague. Before, Ana Luisa did her bachelor's degree in psychology at the Federal University of Paraíba, and then decided to move states and join us here. So that decision has led her into a work relating the respiratory rhythm with some neural oscillations during anxiety-like behavior, which will be the main basis of her upcoming talk. So without further ado, I would like to please give a warm welcome to Ana Luisa.
So, thank you guys. I'm going to start to talk about some statistics related to anxiety disorders. So, 2,064 million people suffer from some kind of mental health disease. Among that, 3.6% represent of this mental health disease is anxiety disorders. Anxiety is ranking as the 6th most debilitating disease. So that's why I think it's so important to study anxiety. But what is anxiety? So anxiety is a state of perception of potential threats that can lead to danger. Represents an evolutionary adapted behavior found in humans and also in animals. Results in different physiological changes. But why talking about anxiety and respiration? So recent studies found that therapies that focus on breathing control show improvements in anxiety symptoms. These therapies like pranayama and yoga. This relationship between therapies that focus on breathing control and the improvements in anxiety comes from the basics that the brain is a part of the body, and they are interconnected in a state of a circular causality. The breathing is beyond a basic life function. So we also know that breathing could modulate brain activity, and maybe that's why the respiration could be so essential in these new studies and as a way to treat anxiety. So the brain, we also know that the brain generates electrical activity, which is closely connected to the firing of neuronal populations. These neuronal populations, they could be described, they could be described like in partners of brain waves. And these brain waves, they are associated with diverse behavioral states. So for example, in humans, we have this theta, alpha, and gamma oscillations, but also in animals. So for example, in rodents, we have theta 2 that could be related to locomotion, and theta 1 that could be related to locomotion, and theta 2 that could be related to anxiety. But why I decided to make this study with animals? So animals models is very good to study this relationship between brain waves and respiration, because we could have a more like inside view of the brain. But how do we do that? So we record respiration simultaneously with the brain activity. So the point is here. Oops, sorry. 2 points. Sorry. What? Thank you. So we have respiration and we also have the brain activity. Thank you. So that's why we could record these both activities and see these respiration coupled oscillations. But again, what is respiration coupled oscillations? So these respiratory coupled oscillations, they are brain oscillations generated by the airflow into the nasal cavity. They are modulated by various factors such as state and behavioral context. So I want that you guys understand that these respiratory coupled oscillations, they are not the activity of respiration itself. In fact, it's a brain activity, a brain oscillation that is generated by the airflow into the nasal cavity. These brain oscillations, they could be characterized by two analyses. So the power spectrum analysis and the coherence analysis. I don't know how much you guys know about electrophysiology, and so I want to make a presentation that everybody could understand. So I'm going to explain some basic concepts, but very quickly, just so everyone could follow. So this power spectral analysis is analysis in which we decompose a signal, electrical signal, in different frequency ranges, and we calculate the intensity of the signal in here related to its frequency. So another thing that is important to know about the brain is that slow oscillations, they have a higher power compared to fast oscillations. Another analysis that is very crucial to characterize these respiratory coupled oscillations are the coherence analysis. This analysis is a statistical analysis that serves to measure if a constant phase difference between any two signals. So we have two signals in the same frequency, and we want to investigate the phase difference between these two signals. So these RCOs, they have been found in different species. So here's a work that is also from our lab, and we can see they collected a data set and analyzed it, and they saw that these RCOs, they have been found in mice, rats, cats, and that these oscillations, they are so important to study because they range in a frequency and similar with other brain oscillations, with other commonly brain oscillations. So for example, delta and theta oscillations. So we started to understand that these oscillations, they are global, but they are often overlooked, as my supervisor already told. So as we can see here, we have this power spectrum and this coherences analysis where we can see this OB is like a proxy of respiration and is compared to the frontal brain, so the frontal cortex activity, and we saw these respiratory coupled oscillations. So these oscillations, they are global because they could propagate through the brain. So initially, the research that study about respiratory coupled oscillations, they found that these oscillations could only occur in olfactory bulb and olfactory regions. But after some recent studies, 20 years ago, maybe, approximately, and they found that these oscillations, they could appear in the medial prefrontal cortex, in the ventral hippocampus, in the amygdala, and these regions is commonly associated with anxious and fear behaviors and states. So taking a deeper look into these RCOs during these emotional states, Sophie Bagou found that the animals exhibit these RCOs with 4 Hz during freezing behavior. So she put the animals in a box, and when the animals when the animals hear a sound, they freeze a behavior that they stay without any locomotor activity, just like this. And they found that they have these 4 Hz oscillations, these RCOs, also found by this power spectrum, this coherences analysis in this 4 Hz frequency. But another open question remained. stayed because these 4 hertz oscillations during freezing is a behavior that is commonly associated with fear. But we also have anxiety. In anxiety, the animals generally in the brain we found that these animals exhibit these theta-2 oscillations. And these theta-2 oscillations, as I have been found, they may be mistaken with RCOs. So here, Adhikari in 2011, they found that this power of the theta-2 oscillations, they could be stronger when the animals was exposed in 2 tasks that evoke anxiety. So we start to ask, okay, animals breathe in slow and fast frequencies, and when the animals was during anxious states, they tend to respirations. So we start to ask, okay, maybe there is a relationship not only with freezing behavior, but also with anxious states. So based on that, that's why my scientific motivation for this study is that the results that I'm going to present, that the results of the data set that me and my colleagues collected during my master's. So we used 7 Wistar rats here at the Brain Institute in a light dark cycle of 12 hours. So we custom-made electrode arrays. As we can see here, we custom-made these electrode arrays, and we planted these electrodes in the prefrontal cortex of the rats, along with a nasal cannula to record the respiration of these animals. And to compare these 2 signals, and we put these animals in a task that has been commonly associated with anxiety, which is the elevated plus maze. In this task, we put the animal in the center of the maze, and the animal could explore freely the maze, but have 2 conditions. They have 2 closed arms and 2 open arms. These closed arms, they are areas where the animals could stay more protected, and the open arms are more open, so the animal could like follow from the maze. So the rodents, they have innate capacity to explore new environments. So these animals come into a conflict. Okay, I want to explore, I want to see what's going on in this maze, but I also am very afraid of that. So we put these animals in these tasks for 10 minutes, and after 24 hours, we expose these animals to the session 2. And the behaviors that we evaluated in the elevated plus maze was the time the animal spent in the closed arms, the time that the animal spent in the open arms, and also a behavior called strat attempted posture, when the animal is in a protected area of the apparatus and he's observing and stretching its body through a less protected area. So for example, we have here the animal is in the closed arms, but he's like stretching his body, putting his head and looking to see if, the potential threat that could be the open arms of the maze. So now I'm going to present some results of this research. So first of all, we found that the animals tend to spend more time in the closed arms, which is also expected because the animals don't have like any motivation to go to an anxious state. So, but a curious thing is that the animals spend the most time, the same time, independently of the sessions. We also evaluate the total distance travel in the maze for the 10 minutes exposure. So in general, these animals, they are put in the center of the arena. They explore more in the first minutes, and then he stay like quiet in the closed arms. But why I say closed arms? Because when we separate this activity, depending on the position in the maze, we saw that these animals, they explore more the center and the open areas of the maze in the first 3 minutes compared to the closed arms that the animals stay in more the last minutes. We also observed the strat attempted posture of the animals, and we also saw this similar behavior that the animals tend to explore more and stay in contact with anxious areas in the first minutes of the task. A curious conclusion about that is because when we work with animals and with behavior, we thought that multiple exposures to a task could relate in some kind of habituation of the animal to this task. So maybe he could be less anxious in the session 2. But this did not happen in this task, in this data set that we analyze. So after that, we also want to look at the respiration of the rat. And we saw something that is very curious. We saw that these animals, they respirate in this like bimodal respiratory distribution. So we thought that these animals, they have like this type of respiratory rate. They respirate in these lower and in faster frequency ranges. But when we saw this power spectrum, we could have a tendency to found that these animals, they breathe more in the fast respiratory frequency rates. But when we saw this histogram, we saw that it's not like that. So the power was greater for fast frequencies, as we can see here, but it's not due to the animals tend to do that more, but only because of the large amplitude of this respiration. And we also analyzed that the behaviors associated with these respiratory rates. So we discovered that the animals, when they are in the closed arms of the maze, they tend to breathe slower compared to the times that the animals was in the open areas, in the center or in the open areas of the maze. So what we can conclude from that anxious animals, they breathe faster. So after that, we came to another question because, okay, we know that the animals, they breathe faster in anxious states, but we have another variable that is very important to understand, that is the locomotor activity. So maybe these animals, they have this increase in the respiratory rate because of the locomotion. So we need to take a deeper look into that, and we've analyzed the locomotion speed of the animals in relationship with the respiratory frequency. So we found that which is like in the literature is obvious, but we found that when the animals, they increase this locomotion speed, they also increase the respiratory frequency. So we start to wonder, okay, so this relationship between respiration and anxiety is not related to anxiety, but only to the locomotion of the animal. So we look at the position in the maze, that's why when we can infer about anxiety, and we found that, okay, that's so bad, because we found that the animals, when they are in the closed arms, they tend to locomote, to like walk less compared to the center and the open areas. So, okay, maybe this activity could be related to locomotion. But when we did the next analysis, we found that not, because we found that the locomotion speed, although it's important to increase the respiratory rate, it's not by itself the reason why these animals, they breathe faster when they are in the open areas or in the closed areas, as we can see here. So when they are locomoting in 5 centimeters per second or in 20 centimeters per second, the respiratory rate when these animals are in the center and open areas, they are increased. So after that, we analyzed the local field potential activity, the electrical activity of these animals. So we found that in the prefrontal cortex, the area where we made our registrations, that these animals exhibit a higher LFP power at the respiration frequency during anxious states. So we separate the whole signal of the brain, and we divide it in different frequency bands, and we divide it into a slower one and a faster one, similar to what we did in that bimodal distribution of the respiratory rate. And we found a correlation. So when the animals, they breathe more, they also exhibit a higher power in these lower frequencies for the electrical activity of the brain. So, okay, now we need to know if this activity is respiratory coupled oscillations. So we analyzed this based on these two analyses, the power and the coherence analysis, and we found that these animals, they exhibit these RCOs at delta and theta frequency ranges during EPM exposure. But this frequency range was more coherent when the animals was in the frequency range of theta 2 oscillations, when the animals was in the open area, open arms and center. So after that, we also want to analyze the relationship between these slower oscillations and faster oscillations. So we make a phase amplitude coupling analysis to understand the relationship between the phase of these lower oscillations and the amplitude of faster oscillations, in this case of gamma oscillations. And we also find something that is very interesting, because although we already know that respiration could modulate prefrontal gamma oscillations, we didn't know that this modulation could be different depending on the EPM hands-on. So we found that in the closed arms, the animals, when they are in the respiratory, in these RCOs slower, they modulate exactly, in fact, at different gamma oscillations than when they are in the center and the open areas. And also, these slower and faster RCOs, they are modulating these gamma oscillations differently. So we can see here, and also in the modulation index, and we can see also in this data set what I already told. So I have some hypotheses to explain these results. I thought that maybe could be different connections, interconnected circuitries that these animals are recruiting during these RCOs and in these different mental states. So I know it's a lot of things for you guys, but I want to leave with some take-home messages. So first of all is that our results confirm previously suggestions about the need to consider physiological biomarkers to complement assessments of anxiety levels in animal models. So I don't know how many of you guys work with animals, but we need to take a deeper look into respiration, because respiration could say something about the behavior of the animal, and it's very interesting, and we need to use like a possible tool to also say that these animals have anxious-like behavior, like have been proposed with other authors. It's not only us that are saying that. So we also have an open question, because as we didn't record in the hippocampus, we cannot confirm that, but we have an open question that is, okay, we found these RCOs in the prefrontal cortex as the same range as theta2 oscillations. So, okay, these oscillations, they are occurring together, they are different oscillations, so maybe they could be mistaken, they maybe could occur together. I don't know, we need to take a deeper look into that. And finally, maybe this could help us to better understand the underlying mechanisms of anxiety and the potential identify new approach to manage these anxiety disorders. So I want to thank to this I want to thank to this great lab, because this work have been done during the pandemics, and when the institute is moving through here in the house, in the old house, so we also in masks, and I really know that this work could not be done without this amazing team that have been my back. So especially David, Joseph, and Elise, that they helped me to collect the data, but also all the members of the TORT lab for the everyday support, coffee, and all my friends in the institute. So thank you, guys.
Is it on now? Okay. So thank you so much, Anna. It was an amazing talk. I believe we have time for 2 or 3 questions. We are a bit ahead of schedule, and I believe everybody's starting to get hungry at this point. But so, does anybody have questions for Anna?
Very short question. You said that these respiratory coupled oscillations propagate to the brain.
Yes.
Now, why we do not see this in VIII?
I think, I'm not sure, but I think that they have already recorded and they found.
Do you think that this is special for the mice, rats, rodents, and could it be different in humans and primates?
No, in fact, that have been already worked with monkeys and also with humans. In fact, during anxiety, they also found these respiratory coupled oscillations in humans. So
in VIII, I'm saying.
Oh, in VIII.
Like in the visual cortex, because we are so visual. Now, I find this all very interesting. I think that perhaps this is a special case for the respiratory rhythms in the mice, but maybe vision contributes, and it would be very nice to see if the eye movements could somehow, how you say, interact with respiratory rhythms in monkeys and humans.
Do we have more questions for Anna?
Thanks. It was a great talk. Congratulations for your work and your supervisor too. I'm wondering whether the co-modulation that you showed before has something to do with theta oscillation, because as far as understood, during the closed arm of the maze, the animal doesn't walk much, or maybe stretch in attended posture, because during the stretch in attended posture, theta oscillation is strong, right? The animal is being aware. So can you comment whether the modulation, the co-modulation that you found between respiration and oscillation is explained by locomotion?
Sorry. Okay. Okay, so we have this hypothesis that maybe these theta-2 oscillations could be mistaken with these respiratory coupled oscillations, and we did not record in the hippocampus, so we can have like a measure that is theta-2. So we have open questions that, in fact, that I'm intending to respond in my PhD. So we want to compare if, okay, maybe these RCOs, they are like occurring together with theta-2. In fact, Adriano already published a paper that showing the parallel detection between respiratory coupled oscillations and theta. So, but we wanted to separate these two and maybe investigate deeper to see if these RCOs, they could maybe could be mistaken with theta-2, or maybe they could occur together. To theta-1. Okay, so we make this power and coherence analysis. So we saw that these oscillations, they are RCOs. So when we made these comodulograms, this phase amplitude coupling analysis, we consider that this in fact is respiratory coupled oscillations. And in fact, this analysis, the theta-1 is ranging from 7 hertz, and what we found in our phase amplitude coupling is in a slower frequency, so is in 4 hertz. So I don't know. I don't think that could be theta. I don't know if I respond your question. Yeah. Yeah. Yeah.
My next question is whether you consider applying some anxiolytic drugs, for example, to see whether you can increase the
We consider that to take a deeper look into anxiety, but we did not put. But we think that maybe with anxiolytics we have like a decrease in the respiratory coupled oscillations and a more exploratory behavior. But as a scientist, like I didn't make the experiment, so I can't confirm none of that. Only hypothesis.
Okay, I think we have time for one last question.
Yeah, congratulations, nice talk. So I was wondering about the time dynamics in your recordings, right? Because if you put yourself in the place of the mouse, the rat, I think.
Yeah, right.
So you start getting anxious before going to the open arm, right? And then you don't have the locomotion yet. So have you tried to like analyze this moment right before because you have this confusion between the effect of the anxiety and the effect of locomotion, right?
Yes.
But then you have a moment where you have anxiety only and not locomotion yet.
Yes.
So.
No. Can't continue. Sorry.
Yeah. So have you thought about dynamically analyzing your recordings in time for trying to grab these moments where you have an anxiety effect without having the locomotion?
Yes. In fact, is one of the analysis that Diego, that has been collaborated with this work, he already mentions like selects it only periods when the animal they exhibit the same locomotion activity to compare these respiratory coupled oscillations. But it's analysis that we didn't make, but it's a great suggestion that Diego have already told me to do it.
Okay, so thank you again, Anna.
Thank you, guys.
And with that, we close up our morning section of this symposium, and go get some food, go have your lunch, and we'll be back at 2 p.m. for a roundtable discussion about depression.